let's move on to the next talk. Dr. Fraser Ferens will be our next speaker. Fraser trained in biophysical studies and received his PhD in microbiology from the University of Manitoba, Canada in 2019. He's a research associate in my lab in the Department of Laboratory Medicine and Pathobiology and the Department of Biochemistry at the University of Toronto. He is studying the complex genotype-phenotype relationship in pseudohypoxic cancer disorders, namely VHL disease, HIF2-driven disease, and PhD-driven disease, using biophysical approaches, which is quite new in this particular system. The goal of his work is to provide the structural information that will inform the understanding of the underlying mechanisms in these cancer disorders. Okay, Fraser, the floor is yours.
All right, thanks for that introduction, Mike. Just let me share my screen. Okay. Okay, so everyone can see that and hear me?
Yes.
Okay, excellent. So thanks for the opportunity to give this talk. Thanks to the conference organizers. Today, I'm going to be talking about VHL and other emergent pseudohypoxic diseases and some information that we've learned and has guided our research from two crystal structures solved in our lab, the pVHL HIF2-alpha crystal structure, and a sneak peek at an upcoming structure from our lab, the PhD2 HIF2-alpha crystal structure. And so pseudohypoxic diseases are named as such because they, as a main driving factor in these diseases, there is an inappropriate hypoxic transcriptional response that is due to the deregulation of the hypoxia inducible factor, or HIF. And so I know everyone's, many of you are very familiar with this pathway, but I'm going to quickly just run through it here. So HIF-alpha is the main regulated component. Under normal oxygen conditions, HIF-alpha is hydroxylated by prolyl hydroxylase enzymes, or PhDs, at two conserved proline residues in the oxygen-dependent degradation domain of the protein. These hydroxyproline residues are detected by pVHL, which is the substrate conferring component of an E3 ubiquitin ligase, which targets HIF-alpha for degradation by the proteasome. Under low oxygen conditions, of course, PhDs are unable to catalyze this reaction to hydroxylate HIF because they use oxygen as a substrate. And HIF-alpha is stabilized and able to dimerize with HIF-beta, where it translocates the nucleus and can govern the transcriptional regulation of hypoxia genes. Oops. So in the human context, this is a little bit more complicated than that. We have 3 PhD paralogs, PhDs 1 through 3. 1 pVHL paralog, and 3 HIF-alpha paralogs, HIF1 through 3 alpha. And of course, these various HIF-alpha paralogs govern overlapping and unique subsets of genes, adding further layers of complexity to the system. Highlighted in red here are the components of the pathway where we find mutations that are causative of pseudohypoxic disease. So PhD 1 and 2, pVHL, and HIF2-alpha. Of course, mutations in pVHL are causative of VHL disease, which is the most well-studied disease of this pathway. So like I mentioned, these diseases are all caused by mutations to proteins in this oxygen sensing pathway. But they also have very similar disease manifestations, which are partially overlapping. And these include renal cell carcinomas, hemangioblastoma, paraganglioma, pheochromocytoma, somatostatinoma, and polycythemia. Today, I'm going to focus on some work we've been doing with HIF2-driven disease. But I'm going to circle back around to talk about what we've learned about how what we've learned about HIF2-driven disease has implications for the other two diseases. So in HIF2-driven disease, we have manifestations of PPGL, which is an abbreviation of pheochromocytoma and paraganglioma, with or without somatostatinoma and polycythemia. These manifestations are observed to be caused by unique subsets of missense mutations in HIF2-alpha, with class 1 disease being causative of cancer and polycythemia, and class 2 disease being causative of only polycythemia. Class 1 disease has been further subdivided into 3 subtypes, class 1A, class 1B, and class 1C, with class 1A being causative of PPGL, somatostatinoma, and polycythemia, class 1B being causative of PPGL and polycythemia, and class 1C being causative of only PPGL. So these mutations in HIF2-alpha are almost entirely localized to approximately 20 amino acid segments surrounding the C-terminal conserved proline residue, which is hydroxylated by PhD enzymes and recognized by pVHL. And this is located within the oxygen-dependent degradation domain of HIF2-alpha and implies that these mutations are disrupting the regulation of HIF2-alpha. In particular, they're clustered around the proline residue, which is known to be more sensitive to oxygen concentration. So previously, some work from a recent PhD graduate in our lab, Daniel Terade, he was able to crystallize VHL in complex with the HIF2-alpha, CODDa, which is the 20 amino acid segment surrounding that proline residue. And we were able to appreciate that the class 1 mutations occurred in residues of HIF2-alpha, which are in close proximity to pVHL and have high potential to disrupt the interaction due to different types of steric clashes or removal of important interacting residues. Whereas class 2 mutations were localized within this loop segment, which makes very little to no contact with pVHL with the exception of F540, indicating that these mutations would likely be less detrimental to the interaction. And this is, of course, what we observed when we looked at some biochemical and biophysical experiments. So on the left here is an immobilization assay where hydroxylated HIF2-alpha peptides were immobilized on streptavidin beads, and the peptides included the different HIF2-alpha mutations. And for the most part, class 1 mutant peptides pulled down much less pVHL than class 2 mutants, with class 2 mutants pulling down less pVHL than the wild type. And this is corroborated by biolayer interferometry data, where we can observe that there is, along the diagonals here, we see the dissociation constant of the interaction of the HIF-alpha peptides with pVHL. And we can see in blue here, the class 2 mutants have mild to no effect on binding, and the class 1 mutants in red have a more extreme effect on interrupting the interaction. Of course, there's an obvious outlier here, which is A530V. So this is a class 1 mutant or cancer-causing mutation in HIF2-driven disease. And it seemingly was able to pull down pVHL at a similar level to wild type, and the KD of the interaction appeared to be identical to the wild type peptide. Now, if we look back at the crystal structure, we can see that A530V is pointing off into the solvent, and there aren't really many interactions going on with pVHL at this residue. So it sort of makes sense that there wouldn't be a big hindrance to changing this A to a valine at this position. So the hypothesis here was that actually maybe this affects the upstream hydroxylation step of the interaction. And so this is the same blot as the last slide, but this second blot here on the right is the same sort of experiment, but this time unhydroxylated HIF2 alpha peptides were immobilized, and they were in vitro hydroxylated with PhD2 prior to pulling down pVHL. And now if we look at our outlier in question, A530V, we can see that the assay is pulling down much less pVHL, indicating that this is actually disrupting the hydroxylation reaction, and that we need to investigate the hydroxylation to fully understand the impact of these mutations in HIF2-driven disease. So initially, my thought was to look at previous structures of HIF1 alpha CODD in complex with PhD2, since there aren't very many differences in the sequences of the CODD regions of the two peptides. In fact, there's only two non-synonymous differences between the two peptides, mainly this methionine to a threonine here, and the insertion of this glycine residue in HIF2 alpha relative to HIF1 alpha. So one of the things, or I guess something that was causing me some concern though, is that G537 mutations in HIF2-driven disease account for approximately 44% of known class 2 disease cases, indicating that this residue is actually quite important for understanding the disease causing mechanism of HIF2-driven disease. So I first wanted to look at the importance. Additionally, I want to look at the importance of this residue, as is known that there is differential affinity or reactivity of the hydroxylation reaction to HIF1 and HIF2 alpha. So I did this looking at using microscale thermophoresis, using HIF2 alpha CODD peptides, HIF1 and HIF2 alpha CODD peptides, fluorescently labeled PhD2 and an inhibitor of PhD2 to stop the hydroxylation turnover reaction from happening to measure the affinity. And what I observed is that I could see the difference in affinity of PhD2 for HIF1 alpha and HIF2 alpha, about a 3-fold difference, which aligns with previous literature values. And then if I just did single mutations in the peptides, so either taking out this glycine or mutating it to a methionine in the HIF2 alpha peptide, I could see that the methionine, threonine to methionine mutation created this intermediate affinity. But if you just delete this glycine residue without mutating the threonine at all, you actually see the same affinity of that peptide as HIF1 alpha to PhD2. Indicating that the main point of difference between these two peptides is actually just the glycine, and then this plays a non-additive, has a non-additive effect on the binding affinity. So I decided to go for direct structural information, and I crystallized the complex of the PhD2 with HIF2 alpha CODD, solved to 1.8 angstrom resolution. And we can see here the HIF2 alpha peptide occupies, unsurprisingly, the same groove in the PhD2 surface as previously observed HIF1 alpha peptides. And the P531 conserved proline residue can be seen to be coordinated next to the catalytic site of PhD2. So if we compare this structure to previous HIF1 alpha PhD2 structures, we can see that the structures are very similar to each other, with the main point of difference being surrounding this G537 residue and the preceding T528 residue. Here, I'm just going to talk a little bit more about the situation, or the about the differences in this area of the complex, more zoomed in. We can see that this glycine residue here has is incorporated in a sharp turn in the peptide backbone, and this is happening to accommodate the same interactions that HIF1 alpha has with PhD2, with the notable exception that this methionine 535 is more distant to the tyrosine 403 on PhD2 relative to the HIF1 alpha structure, where the corresponding methionine is a little bit closer. And this indicates that this interaction here would be slightly weaker. And this is likely the source of our differences in affinity of the two peptides to PhD2. Also, the relevance of G537 to HIF2-driven disease becomes apparent as this sharp turn here causes the glycine residue to adopt fine psi angles, which are likely unfavorable for any other amino acid, which when this residue is mutated to another amino acid explains why maybe these neighboring interactions can no longer be maintained and why there would be interruptions to hydroxylation. So if we circle back around, now it's talking about class 1 mutations. Class 1 mutations are localized here to residues close to the active site of PhD2 and to distal anchor points on the interface. And we can see that the mutations correspond to residues which are making interactions that are necessary to coordinate this P531 into the active site, or some mutations occur on P531, which of course, if this proline residue is missing, disqualifies the peptide from being hydroxylated. And this is true too of the more distal sites. So here we can see L542 is making hydrogen bonds with neighboring PhD2 residues, and the side chain is occupying a small pocket on the PhD2 surface. Meanwhile, class 2 mutations are observed to be localized to the more middle region of the peptide, and this region was found to be much more flexible than the rest of the peptide in the structure, and this is evident from the elevated B factors of this region of the peptide, indicating that the interactions between PhD2 and HIF2 alpha in this region are less strong than the interactions surrounding where we find class 1 mutations. But we also don't need to only predict the effect of these mutations on the interaction. I can directly measure them. Using the same method that I used before, microscale thermophoresis, we took the class 2 and class 1 mutations that were previously assessed in our study of the pVHL HIF2 alpha interface. And basically, yeah, we see that it segregates by disease class, with class 2 mutations having a more intermediate effect on the binding of HIF2 alpha to PhD2, and class 1 mutations having an extreme effect with much, much higher dissociation constants, and also many of them I observed no binding between the two in the concentrations examined. So for HIF2-driven disease, we can come back then to a model where class 2 mutations have an intermediate inhibition of the hydroxylation reaction by disrupting the affinity of HIF2 alpha to PhD2 or other, and likely other PhD enzymes. And then having a mild to no effect on the recognition of hydroxylated peptide by pVHL. And this should lead to some stabilization of HIF2 alpha over wild type levels. And this leads to polycythemia. Class 1 mutations, on the other hand, have an extreme effect on the interaction of PhD2 and HIF2 alpha, likely disrupting hydroxylation quite a bit. And also have a more extreme effect on the recognition of the hydroxylated peptide for, or the hydroxylated protein for by pVHL, which would lead to much higher stabilization of HIF2 alpha, which would result in the more extreme cancer phenotypes that we observe in class 1 disease. So what does this mean for pseudohypoxic diseases in general? So we can see that the different classes of HIF2-driven disease seem to be caused by varying disruptions to the regulatory pathway of HIF2, which should result in varying degrees of stabilization of HIF2 alpha. And then these varying degrees of stabilization have been shown to be causative of polycythemia, PPGL, with or without somatostatinoma, and in 2 cases, I believe in the literature, hemangioblastomas, which we also observe as disease manifestations in type 2A, type 2C, type 3 VHL disease, and also with PhD2 mutants, meaning that likely these manifestations are driven by a gradient of stabilization of HIF2 alpha, with the odd manifestation out here being renal cell carcinomas, which aren't observed at all in HIF2-driven disease. Although I think from the discussion earlier today, there is some talk about, is HIF1 alpha also involved in RCC? Are there other players involved? I think there's a lot of room for further study in this area. So with that, I would just like to take a moment to acknowledge a few folks. So big thanks to my supervisor, Michael O, and our close collaborator, Jeff Lee, also at U of T. Daniel Terade was the PhD student, now graduate. So Dr. Terade worked on the pVHL HIF2 alpha interaction story. The members of the OLAB for helpful comments and discussion throughout. And of course, thanks to Beamline 17, ID 1 and 2 at the NSLS2 for the X-ray data collection, and of course the funders, CIHR to Mike, and the Canada Research Chair Program and the Canada Foundation for Innovation to Jeff. And I will take questions.
Great, thank you, Fraser. Very detailed analysis, structural work, quite impressive. Yeah, Osan has his real hand up, so.
This time. No, this is great work to break down, the linkage of molecular changes with the phenotypes. I'm wondering every time, and this is a question for you, Fraser, and from everybody else, every time that we start thinking along these lines, I'm wondering, why not RCC in HIF2 alpha germline mutations? It brings back again the point that it's something else VHL is doing besides RCC to create renal cell carcinomas, despite the fact that the classic phenotype genotype correlations, we remember this paper from Bill's lab, indicate that the risk for RCC is proportional to the inability of VHL to bind HIF.
Yeah, I don't know if I have a good answer for that. I have speculation, perhaps. I know that there are recent animal models that show that both HIFs are required for RCC tumor initiation, but that HIF2 is more important for tumor growth. But yeah, I think that, it's very possible that there, I guess, are other functions of VHL, or that other HIF activity is necessary to explain RCC, such as HIF1 dysregulation in addition to HIF2, which in HIF2-driven disease, of course, we only see the deregulation of HIF2, which is, I guess, a little bit, I mentioned inappropriate hypoxic responses. I think that's even more inappropriate than the, I guess, situation in VHL disease where both HIFs are deregulated. But yeah, I guess it's just an interesting area of study where we need to do more work.
Yeah, and I think if I can just chime in a little bit, Fraser, we've heard talks today about, RCC and other prominent genes, the PBRM1, SETD2, BAP1. So there are definitely, I think, other players in addition to HIF1 in early event. For example, during the cyst development, there is a prominent role of HIF1. So you can imagine in sort of an experimental setting where knockdown of HIF2 would regress these xenografts, because we're talking about fully grown tumors. But in the actual human development of kidney cancer, I think is far more complicated. And I think the genetic evidence is pretty clear with the HIF2-driven disease. We just haven't seen it. So I think HIF2 alone probably cannot initiate it, but once it's initiated, it's clearly important. So that's roughly where we're at with that thinking, Othan.
Yeah, that's very interesting. Thanks, Mike and Fraser.
There's a question in the chat that I can read out if there's time.
Yes, please do.
Okay, so from Patricia Dahia, she says, thank you for the interesting talk, and asks, what are your thoughts on the fact that there is overlap between the genotype of PPGL alone versus PPGL with somatostatomas and or polycythemia with respect of the EPAS1 mutation? Instead, they may occur at different times during development, mosaic versus somatic developmental window.
Yeah, I would say that is certainly possible. I will preface this by saying that I am a sort of hardcore biochemist, biophysicist. So the cancer biology part of it is sort of what I'm learning right now in Mike's lab. So maybe I'm not the best equipped to answer this question. But I would say that, yeah, that does make sense to me. And that, I would sort of liken this to some of the disease progression we see in class 1 disease, where we, in the instances where we have patients that are presenting with both PPGL and polycythemia, they always develop polycythemia very early on in life. And then we see decades later the development of PPGL. And so I think that, yeah, there would be some sort of temporal link too in some of the mutations.
Thank you. Is there time for a quick follow-up on this?
Absolutely. Go ahead.
Yeah, you may have mentioned this during our talk, but I'm always curious. I'm from the PPGL world. What happens to proline 564 and why is it never mutated in these tumors, or none that we have come across so far?
Yeah, so so are you referring to in HIF1 alpha?
HIF2 alpha.
HIF2 alpha.
HIF2 alpha.
In HIF2 alpha, it's P531 that's mutated. Are you referring to
Sure.
P405, the earlier N-terminal?
402. Sorry. 402.
Yeah, okay. Yeah. So this, yeah, I think is probably more related to which genes each HIF alpha isoform regulates in the hypoxic response. I think that a lot of the differences there, like we don't see it because the regulation, the subsets of genes that each isoform regulates may not have the same effect in developing the cancer phenotype. I mean, for instance, we don't see any, as far as I'm aware, mutations of HIF1 alpha that are causative of these tumors. Yeah, my best guess would just be that it's down to which genes downstream that they regulate, and the ones that HIF2 alpha regulates are more oncogenic.
There's just one more question from Carol Pacat. Any view about why do somatic gain of function EPAS1, i.e. HIF2 mutations exist mainly in females?
This one I feel very unequipped to answer. No, I do not know.
It is an interesting observation.
But it's an interesting observation.
Yeah, we are a bit of at a loss with that one. More work to be done. Thank you for that question. Okay, thank you, Fraser. Let's move